Now we'll move to our second speaker, Dr. Richard Pyles. He's a professor of pediatrics, also professor of microbiology and immunology at the University of Texas Medical Branch, UTMB. As director of the Galveston National Laboratory Molecular Diagnostic Lab, his group performed clinical COVID testing. His ongoing research includes exploration of the role of the nasal microbiome on SARS-CoV2. The work has discovered antimicrobial bacteria that help identify novel antimicrobials and molecular pathways to limit the virus. So, Richard, would you take over, please? I see you just started. Thank you.
And you can hear me all right?
Yes, I do.
Very good.
Thank you. The floor is yours.
Thank you for the introduction, and thanks also to the organizers for putting together what is really remarkably diverse and interesting symposium. A really nice way to spend a rainy Wednesday morning. Ultimately, I'm here to present our story, and I'm very proud to be the individual that was asked, but I need to acknowledge the fact that this was a really huge group effort. It will be a blend of both clinical and research efforts that I share with you today. I want to acknowledge in particular the colleagues of mine at the Galveston National Lab, Carrie Maxwell, Aaron Miller, and Nigel Bourne, came in tirelessly every day while everyone was sequestered at home to execute lab-developed testing that I'll tell you more about. We also were not able to do this without the support and constant guidance of our clinical side of the house. Over here, Juan Garcia, Ping Ren, Natalie Boyer, and Mike La Posada really created the opportunity for us to accomplish this work in response to this pandemic event. Finally, I do want to acknowledge the WRCEVA that provided us with the original material that allowed us to bring these assays online very quickly, as well as the research folks that keep us safe and the environmental health and safety team. Finally, funding was provided by a number of organizations that are really very special scenarios in Galveston County, and with UTMB in particular, we have benefited greatly by the Seeley and Smith Foundation. The fact that we have a local CTSA award that has established the Institute for Translational Sciences, and a university president, Dr. Raymer, who's very willing to write a check to keep this work going. So as I said, this was a real collaborative effort that brought a research lab into the clinical arena. You can see here some images from the lab that show our needs for attaining a research level of safety compliance that required us to wear PAPRs and particular PPE to process these clinical samples. That's unusual on the clinical side of the house, as they have essentially different set of regulations. So in this hybrid event, we actually had to be compliant with both research and clinical aspects of compliance and regulation. To accomplish this then, we had to bring laboratorians, diagnosticians, and researchers together in a very quick set of meetings to make sure that we were compliant on both sides of the house. This hybrid event really grew from our past responses that had been mentioned in past pandemics. And that prompted me to think about a slide deck that I put together for describing what we did during the H1N1 swine flu event in 2008, 2009. And I have taken one of those slides and added it to this deck because I thought it was really relevant and remarkable that the questions have remained the same. So in this H1N1 swine flu event, there was a novel flu that came out of Mexico that was missed because of limited surveillance. That was a herald to the pandemic. And ultimately, after very careful molecular characterization, the H1N1 was defined as a reassortment of avian flu and swine flu. So questions we had at the time, again, are remarkably similar to those that we're experiencing with SARS-2 now. We had some theories back then about where the virus originated from and how it moved to humans. But we identified back then a new word was coined and put into the lexicon, the infodemic. That I think remains a real impediment in some ways to the effective testing and processing of all this material. So with this information in mind and our past experiences, in February, we began the process of responding for the COVID-19 event. We brought lab-developed testing to the forefront, and this again was made possible because the WRC-EVA was able to provide us with extracted RNA from the original WA1 CDC isolate. With that positive control material, we made modifications to both the CDC and WHO qRT-PCR assays and selected targets that you can see here for both the human and the viral sampling. We established the workflows working very closely with UTMB clinical groups as the time since the swine flu has seen a real progression in information technology that changed the methodology for sample management, tracking, and reporting. We needed to be brought up to speed, and as a research lab, actually had to import some hardware that allowed us to properly meet the regulations. This was based and was only successful on relationships with clinical micro and molecular diagnostic groups here that drove this bus such that by March, we were able to have the assay performance completely assessed, EUAs prepared and submitted to the FDA that described the sensitivity and specificity. They mobilized from their archive about 100 NP swab samples that were collected pre-pandemic, but coincident, so February in 2020, that were positive for other respiratory pathogens, but negative for SARS-2. Using those samples, we proved our sensitivity, our specificity, and then did confirmation in silico as well. All of this work collectively brought us to the forefront on March 19th, and we scaled up with the intent of providing not only initial testing until commercial platforms could be brought online, but the ability to provide surge backup when those commercial platforms became overwhelmed or, as we discovered right after initiating testing, compromised by the supply chain. In April, in fact, we already had evidence that the supply chain was going to cause us problems with our established assay, prompting us to begin the process of developing a labor-reduced, material-reduced alternative that we termed the BP assay. This was essentially back to my graduate days. I won't share how long ago that was, but it utilized a boiling method instead of a full extraction method. There are a number of papers in the literature now describing this approach, which becomes much less dependent on a robust supply chain. So in April, we fell short of our planned mark for capacity, but we're able to bring this second set of assays on. Over the course of this experience, my lab group has been responsible for completion of 27,000 charted results and over 30,000 actual assays. That distinctive difference, about 3,000, provides me an opportunity to tell you some of the additional stories that were going on that again echoed the concepts of supply chain fails. It wasn't just about the molecular reagents, but also about the testing kits. And as the COPAN and BD products became shorter and shorter in availability, we reached out and started looking for alternatives. A company called Medico out of China advertised a surplus of these transport kits that ultimately were purchased by Alibaba.com ordering. This is essentially, for those that aren't aware, an international Amazon, if you will, which is not meant to be offensive. Early on in the implementation of these testing kits, we had our first run come back with some real terrifying moments. You can see here that after the inactivation bleach solution was added to a BD, we saw the expected color change, confirming that this material had been inactivated and was no longer a biohazard. As an alternative, the Medico, you can see, did not turn colors. That led us to reevaluate our process, determine whether or not the bleach was somehow not properly dispensed. But in the end, it turned out that Medico had elected not to use the pH indicator phenol red, but rather a red dye. And that red dye caused us great consternation. Another example of reaching the limits of the supply chain, Premier Medical in Texas was a service provider that decided that they could produce NB kits using standard 15 mil conicals. They decided, in the absence of available antimicrobials in phenol red, to modify the standard method. The net result here was the production of mold. And in samples that had already been collected, mold was detected, leading our group to step aside for a period of time to actually do some validation with spiked versions of both the raw Medico and Premier Medical NP swab kits, showing that they produced results that were equivalent to the Copan or BD kits. This allowed us to salvage all of those collected materials that had actually grown mold. So with all of this work and the commercial platforms coming online roughly in May, we worked our way through a July surge that many others saw, and collectively, as of the end of April this year, UTMB had completed a combined testing of over 400,000 samples. I say that with great pride, as having only contributed about 27,000 to that number, and reminding everyone that we're based in Galveston County, where the county population is only around 350,000. So I applaud my colleagues for the massive effort to respond to this pandemic and really bring these testing opportunities to the forefront of our efforts. This work also led us to have research potential. One of the first studies that we completed utilized the MDL's approach for environmental swabbing that was required to be research compliant. And that allowed a methodology for environmental sampling that we applied to an operating room that saw a localized outbreak for operating room staff. And this was published recently. Lead author April McDougall really brought together, again, a really interesting group of clinical and research staff here at UTMB to accomplish a really careful evaluation of how this outbreak occurred and whether or not our efforts to keep these types of facilities clean had achieved their goals. Another opportunity that was presented by our effort was to really look at the viral titers. And one of the claim to fames that I think will go down in the record books for this particular virus is the remarkably high titers that were found in clinical samples. In my 30 years of research experience, I had never seen positive samples that were this hot from the clinical arena. So we did a variety of evaluations. Here I show you a violin chart by decade of life across the samples that were collected in July. And what we immediately recognized was there was really very little difference in age contribution for this particular sample set, with the small exception of the less than 1-year-olds that were included in the cohort having a slightly average higher titer, but this was not significantly different. It really laid the groundwork for our thinking about why this range of viral titers might be different. And now that we have been able to back down our testing bandwidth, we've begun to re-engage our research aspects. And in this process in July, where we had this peak, we started to recognize clinically this concern that was shown here in a snippet from an Excel spreadsheet. So on July 9th, here are the ORF8 results for CT. This is the return from a PCR assay versus the envelope. These numbers should be relatively similar. And then the housekeeping gene to show whether or not the sample had adequate quality. We began to become concerned that our e-assay was beginning to expire because we saw a negative sample. It's not unusual. The response in a typical clinical lab is to eliminate those reagents and replace them with fresh materials. We did another batch that day and all of a sudden began to see this E dropouts with greater frequency. As of July 10th and 12th, with additional runs, we saw additional samples. So having replaced the reagents, we really felt this might be indicating that there was a change in the viruses. And this was in advance of all the variants actually being shown. So we went back and looked at the specific locations and potential consistencies that these samples provided and found that they were all from what was essentially an outbreak in one of the prison populations that UTMB was providing testing for. There were about 40 samples that fell into this category and were very restricted to not only the location, but this time frame. So this was not a variant that essentially escaped the prison population and led to common circulation. And this was identified simply by lack of detection with a PCR product. Another opportunity that this provided us was based on the fact that the WRC-EVA at UTMB is charged with retaining a repository of strains from all over the planet. We decided we would be able to contribute to that. So beginning on March 19th, the first day of testing in 2020, all the way through January 6th, we selected interesting NP swab samples and transferred those to the Enhanced Biosafety Level 3 facility, where Barbara, Judy, and Tom Kizak aggressively pursued isolation of the virus. This was a work in progress as there were a number of things that we had to address to enhance our success. But out of 201 attempts, 116 distinct viruses were isolated. These were then sent on to the UTMB pipeline of Andrew Routh and Scott Weaver, who were doing full genomic sequencing, looking for changes in both the outgrowth and the original material that we were providing. This prompted Andrew Routh's group to create a new method for NGS that was higher throughput and lower costs, called tiled ClickSeq. This really has potential for providing almost a shotgun approach to the identification of novel viruses coming up in the population. So I'd encourage those that are interested to take a look at this publication. This effort ultimately also took us back to the idea that there were 8 logs of difference in titer over the samples that we evaluated in July. We really sat down as we had more bandwidth for research and asked some very basic questions. I've shown you the break apart by age. Here's the gender results. We also looked at comorbidities, socioeconomic diversity, and ultimately came to the conclusion that a lot of this noise was potentially created by the fact that people went for testing at different points in the viral infection process. Some of them were symptomatic, some of them were simply concerned, some of them had been contacted to be told that they had an exposure event, leading to sampling early, in the midst of the highest titer, or even late. And that really led us to the final question that powered the rest of the talk today with regard to the research, and that was how to best define what was a high titer versus a low titer infection by eliminating a lot of this noise. So we used a cutting edge technology many of you are familiar with called RNA-seq directly in the clinical swabs. And we decided that we'd sort of turn this concept on the head of rather than looking at the virus, look at the human's response. So we analyzed the expression pattern of over 27,000 human genes, and then using unsupervised clustering, ask whether or not this approach would produce a cluster that was representative of high and potentially low titer infections. We were very pleased to see that this hypothesis was proven correct, at least for the high titer. And when we drilled into this red box of 6 samples, we found these to be the highest titer samples that we analyzed by the RNA-seq approach. So by 2 different methods, we had essentially proved that this concept was good. So we took the average titer for these, and then in the blue boxes, the average titer for the low, and decided a cutoff for high-low demarcation was going to be best set at 5 times 10 to the 4th viral copies. That then becomes the basis for the rest of our work. Using that cutoff, we went back to some clinical samples. Male and female didn't really shake out as dramatically different, so we asked a more straightforward question about the viral receptor. Was transcription level of the 2 main receptors different based on high and low titer? We found that was true for ACE2, but not for the TMP. And ultimately, this significant difference really powered the idea that we were on to something, but it wasn't just about the host. when you consider the main site of infection as the nose, one of the interests in my group has been consistently the nasal microbiome, or the bacteria that are found in the nose, commonly very diverse across different racial profiles, socioeconomic profiles, gender, etc. And so we ask again, using this high-low titer demarcation, if we could begin to distinguish differences that were either caused by the presence of the virus or set up susceptibility to the virus. We made a very small pilot cohort, and with funding from Seeley and Smith, we evaluated the titers of particular bacteria of interest. Corynebacterium as a genus is commonly considered a probiotic in the nose. Pseudodipteroticum was a particular species that has been associated with some antiviral properties in other systems. And what we found for Pseudodipteroticum was a much higher titer of this bacteria in the context of low titer samples, suggesting there might be some connection here. Another bacteria that's common to many mucosal sites are the Staphylococci, in particular Staph epidermidis is another known antiviral bacteria. And so we looked specifically by PCR for those outcomes as well. And we're heartened to see that there were distinctive differences, but in this fairly small cohort, we didn't achieve significant differences. So while these associations were very intriguing, they didn't allow us to really do any kind of causation or direct evaluation, which brought us to this model system that we reported a couple years ago that was essentially built to culture the nasal mucosa in a way that would support colonization by transplanted human nasal microbiota. I won't refer to much of this paper for the sake of time, but hit the highlights of the way this model works. It essentially utilizes what's called a transwell filter, which is an insert in a culture vessel. The transwell filter allows a porous membrane platform onto which the nasal epithelial cells are plated. After that stabilizes, we remove the culture medium, and I have to give full respect to my wife's lab for convincing us that we should try this. It's in the marriage contract at this point that I acknowledge the work from her group that didn't lead to the death of the cells, which was what I said was absolutely going to be the case. After removing the culture medium, these cells actually matured into differentiated mucosal and actually began to produce an environment that was capable of supporting this robust microbiome. This model system shown here in scanning electron microscopy, you can see the diversity of cell types with lots of globule producing, even some evidence of some matrices that ultimately at a lower magnification can be observed as potential mucin globules that ultimately are responsible for production of mucus. Contrast those sterile culture images with those that have been colonized by a nasal microbiome, and you can appreciate the presence of the distinct morphotypes of the bacteria. And in these two images, you can appreciate the formation of biofilm. So in a very biological fashion, we validated that the environment created was able to support these complex communities taken from a donor's nose. So the basic workflow for these studies was then to create the multi-layer using a male culture and a female culture, and then ultimately challenge them with SARS-2 in a BSL-3 environment, with or without the addition of a nasal microbiome. Initial work was done simply to determine whether or not these cultures would support the WAL1 CDC isolate. And in fact, it did. 72 hours after infection at two different multiplicities of infection in both the male and in the female produced very robust replication of the virus. We also were able to look in this polarized chamber system at what would be essentially the systemic compartment and whether or not the virus was released into the basal chamber, and it was in fact. So with this preliminary data, we were able to identify a methodology, and this was in part published in the Zika event for a vaginal model system where we actually looked for vaginal microbiota that would impact Zika and herpes simplex infection. So the details of this approach can be found in that publication. When we took staph epidermidis, a nasal microbiome number 40 that contained Carini, Pseudodiphthereticum, and staph epi, and compared that to nasal microbiome 42, which lacked both epi and Pseudodiphthereticum, we took this through our very standardized workflow, added SARS, and as the main outcome, asked whether or not these bacteria had an impact on the viral replication. And I'm very pleased to tell you in this preliminary study, we proved that the model would work to begin to address the way that these bacteria might impact in the real world an outcome. With all of this together, we were able to essentially put together several grant applications. And with that, I will simply note the future directions and the continued effort of my group to provide surge and assay development for COVID testing needs. And then a long list of things with this culture model that ultimately, I hope, will lead to novel intervention developments in the form of nasal sprays. As I mentioned, this was work that was done as a dramatically impressive team of clinicians, pathologists, and researchers. And again, I note the funding and appreciate the opportunity to present this work.
Thank you very much, Richard. And also thank you for this overview of your clinical work and your research, which is a very good example of a translational research project, sharing ideas, instruments, models between the lab and the clinic or patient data. So we have a few questions. Let's start with, are there nasal microbiome profiles shifting with the SARS-CoV-2 variant?
So we have not had a chance to evaluate any of the variants. Our research bandwidth has been fairly narrow as we've been focused on clinical testing, but as this has lightened up, we've been able to begin addressing that. We're excited for that opportunity. At this point, all the work we've done has been with the original WAL1 isolate from the US.
Okay, another question is, what would be the impact of antibiotics on the nasal microbiome?
Back to an add-on to that I would suggest is interesting from Dr. Domenici's talk earlier, the addition of air pollutants. We're modeling with the use of cigarette smoke extract in this culture model. So both of those have dramatic effects, especially on the development of biofilms. And those biofilms, we think, are going to be additional physical barriers to the virus reaching target cells. And this is a really interesting area for us. I wish I had a more advanced answer at this point, but it's early in our pipeline. We have done antibiotic evaluations and other studies, and the bacteria are actually remarkably resistant to the presence of either a topically applied, almost an ointment or a nasal spray, versus a systemically delivered, in that case, the antibiotics placed in the basal chamber. The bacteria that fall out are often not the pathobionts that we're most concerned about.
Okay, and sometimes it is found that actually smokers seem to be protected from the infection, which is from a health policy, some kind of a disaster, but do you confirm this finding as well?
I can't tell you that we have or we haven't yet. The work's not completed, but we're excited to get to that kind of an answer.
Okay. So maybe just one short question. So you talked about the supply chain, how it failed, and how regions could be interchanged, but with major consequences in terms of testing, etc. Are there also other substantial challenges when you are running a lab performing tests during the pandemic? Just
thanks for that question. Yeah, certainly this is a very fluid scenario. I can give you two examples in this regard. in the response to the swine flu, we learned a lot. We were prepared, we thought, for the next event. But in the 10 years that had passed, CDC, FDA, a number of additional regulatory bodies had changed the goalposts and led us to essentially restart much of the regulatory compliance issues. Secondly, as the supply chain's fluidity impacted everyone's ability to execute this testing, the FDA changed the guidance on what they expected as the minimum data set for successfully competing in an EUA. That led us back to the drawing board multiple times to create additional data sets to then share. We were in real time working with other sites in Louisville and Alabama and a number of other network labs, and we're trying to share information. And as it turned out, because of the restrictions on even the PCR platforms that were being used, we actually kept going back to the drawing board to try and revalidate. That added not only a lot of labor to the process, but took out very critical supplies from the actual testing pipeline. So I think those are lessons that we will be able to apply better, hopefully, to the next event.
Let's hope so. Yes. Thank you very much, Richard, for your speak.